This will be a walkthrough of the AQA GCSE Biology 2022 Higher Tier Paper 1.
Okay, question 1. This question is about cells in transport. Complete table 1. So table 1 is showing us some parts of a cell, which some are missing. It's also showing us the function of some of the cells, which we don't know the functions of, or we don't know the names of. So first one, does contain genetic information. Well, that should be the nucleus. Second one, mitochondria, so we need the function of the mitochondria. Well, there's a variety of different answers we can put here. Main one that we should know is it is where respiration takes place. However, you could also write it's to release energy. Okay, and last one, controls the movement of substances into and out of the cell, or should be the cell membrane. Cannot be the cell wall. The cell wall is support. Okay, next question. Cells in potatoes are plant cells. Cells in potatoes do not contain chloroplasts. What is the function of chloroplasts? Again, multiple answers you could put for these. Main one we should be saying is where photosynthesis takes place. But you could also say it's to absorb sunlight. Next question. Name one type of cell in a potato plant that does not contain chloroplasts. So we need a cell that's not going to carry out photosynthesis. So thinking about a plant, these cells that we find in the roots of the plant are called root hair cells. Another one which you may put, remember it's only asking you to write one, so you should only be putting one, but other options could be the xylem. Okay, next question. A student investigated the effect of salt concentration on pieces of potato. This is the method used. So we should be reading the method carefully and trying to ensure that we can think about the variables that may be in this investigation. Independent, dependent, and control variables we all should be able to identify from reading the method. So first question, 1.4, what is the independent variable in the investigation? So we need to remind ourselves what independent variable means in an experiment. It means to change. So reading our method again, what would they be changing? It says repeat steps 1 to 6 using different concentrations of salt solution. So if you look at our options, concentration of salt solution, first one, that should be the mark. 1.5, why did the student dry the surface of each potato piece with a towel in step 5? So we're going back to step 5, dry the surface with a paper towel. Well, if we take out our potato pieces from our solutions, they're going to have some water on its surface. And we don't want to measure that water or solution because that will affect the reading on the balance. So it should be to remove excess water. Should be able to get away with solution. However, why do you want to do that? Well, if water or solution remained, what would happen to the reading? It would be higher. At 1.6, the student calculated the percentage change in mass of each potato piece. For one potato piece, the starting mass was 2.5 grams, the end mass was 2.7 grams. Calculate the percentage increase in mass of the potato piece using this equation. So it's great that they've given us the equation, so we just need to find out from these values where they should go in our equation. So starting mass is 2.5 grams. Our increase in mass needs to be the change between our two masses. So it should be 2.7 minus 2.5, which is equal to 0.2 divided by 2.5. And then you need to times that by 100. So we should come out with a value of 8%. Okay, the student used the results from each potato piece to calculate the mean percentage change in mass at each concentration. So here is our result. Our independent variable, definitely concentration of salt solution on our x-axis, that should be on our graph, and our mean percentage change in mass should be on our y-axis on our graph. Complete figure 1. You should label the x-axis, use a suitable scale for the x-axis, plot the data from table 2, and draw a line of best fit. So it's given us the different points that we need to ensure that we can get 4 marks. So the first thing that we would need to do here is we need to label our axes. So what our axis should be is concentration of salt solution in moles per decimeter cubed. Okay, then we would need to put a scale for our x-axis. Remember that our scale should be even, so it's starting off at 0.0, then we should be going evenly 0.1, 0.2, 0.3, 0.4, 0.5. So realistically we're going up 0.1 each time. We then are able to plot our points. So we look at the table carefully and then we make sure that we plot our points. We make sure that we understand how much this scale is going up in on our y-axis. You can see it should be going up 0.2 each time for each line. So first one, 0.0, finding 9.8 on our x-axis and our y-axis, and then we put either a dot or a cross there, making sure it's quite visible. 0.1 should be going to 9.5, so we would need to put a little dot just there. 0.2 going up to 7, 0.3 to 0.4, and then on 0.4 we're going to negative minus 1.4. Okay, so our points are plotted. We now need to provide a line of best fit. Now, what we should be doing in a line of best fit is looking at the shape of the points and then creating a line which fits most of that data. So in this case, you can see that our line is starting to form some sort of downward curve. So we should not be trying to do a straight line of best fit, because it will not go through any of the points if we do that. We should be looking at the pattern that the points are and then creating a line which fits that. Our line should also be smooth. We should not be using a ruler. So as smooth as possible, take a pencil and make sure you do your smooth curve through the points. Question 1.8. What concentration of salt solution was equal to the concentration of solution inside the potato pieces? Well, our experiment is investigating osmosis. So if we remember, osmosis is the movement of water molecules from a high concentration to a low concentration through a semi-permeable membrane. So in this case, we want to find out a situation where the concentration is equal inside and outside of the potato pieces. So that would be causing no change in mass. So we would need to find where does our line hit the x-axis at 0.0. Looking carefully, it should be around 0.31 or 32 from our curve here, and that should be our answer. Question 1.9. Explain why the potato pieces in the 0.4 moles per decimeter cubed salt solution decreased in mass. So we need to find our table again, or look at our graph. Our percentage change in mass was minus 1.4. So that indicates to us that the water had left the potato. We have to remind ourselves again that this investigation is about osmosis. So water molecules left the potato. Why would the water molecules leave the potato? It's to do with the concentration gradient. Remember that osmosis, water moving from high concentration to low concentration. So that must mean that the solution contained a lower concentration of water. But to make sure that we demonstrate our understanding of this investigation, we should be writing how this is occurring. Water molecules are leaving by osmosis. Question 2. Plant cells and fungal cells are similar in structure. Figure 2 shows a fungal cell. So if we remember our fungal cells, this should have a nucleus. Name one structure in figure 2 which is present in both plant cells and fungal cells but not in animal cells. So we need to think about how these are similar but different to an animal cell. When looking at the figure, you can see that there are two borders on our fungal cell here. The outermost border has always got to be a cell wall, so our animal cells don't have a cell wall, and that should be our answer. Question 2.2, which disease is caused by a fungus? Okay, so we're thinking about infection and response, and our diseases that are caused by our different pathogens. So gonorrhea is caused by a bacterial cell. Malaria is caused by the protist. Measles is caused by a virus. And rose black spot is our plant disease that's caused by a fungus. (2.3) A fungal cell divides once every 90 minutes. How many times would this fungal cell divide in 24 hours? So we need to think about how many cell divisions are going to occur now. So we have two time periods, one 90 minutes and one 24 hours. What we would need to do to try and compare them is convert our time period. So the easiest thing for us to do is to convert our 24 hours to minutes. So if we remember that 1 hour contains 60 minutes, we should then try and figure out if we have 24 of those hours, how many minutes that would be. We would then need to multiply our 60 by 24. That should be 1,440. So we now know how long 24 hours is in minutes. We now need to think about how many divisions are going to occur in this 90 minutes. So we need to take that 1,440 and then divide that by 90 to find out how many times it's going to divide. And the answer should be 16 cell divisions. Question 2.4. Some types of fungal cell are grown to produce high protein food. The high protein food can be used to make meat-free burgers. Where is protein digested in the human digestive system? Our large intestine, its function is to absorb water, store our feces. Our liver is supposed to produce bile. Our salivary glands are supposed to be producing the enzyme amylase, which is supposed to break down starch, whereas our stomach is supposed to churn our food and also help to digest protein with the presence of protease enzymes. So the answer should be stomach. 2.5. Which chemical can be used to test if the burgers contain protein? Right, so we have our different types of solution to test for foods. Our Benedict's reagent is supposed to be testing for sugars, so this cannot be Benedict's. Our Burette solution is to test for proteins. How can we remember that? Beep beep. Burette testing for protein. The color changes from blue to purple. 2.6. Table 3 shows some information about burgers made from meat and meat-free burgers. So here are different nutrients that we're going to find in these two types of burgers, and then how much of each do we find 100 grams of burger. Evaluate the use of burgers made from meat compared with meat-free burgers in providing humans with a healthy, balanced diet. Use information from table 3 and your own knowledge. So we need to be evaluating. What must we do when we evaluate? We need to compare. We need to find the advantages or pros. And we need to find the disadvantages and cons. And then we almost always need to provide a conclusion. So we must make sure that we do all of these things in addition to using the data from the table and any of our own knowledge which we know about these nutrients here. So first thing that we want to do is we want to compare. So we go through each of these nutrients and compare. So protein, we can see, is higher in burgers made from meat. There's a comparative language that we use, higher. Fiber is higher in meat-free burgers. Fat is higher in meat burgers. Carbohydrate is slightly higher in burgers made from meat, but we could say it's really similar because it's only about 0.4 difference. It shouldn't make too much. And then cholesterol is much higher in burgers made from meat. So we've done our comparison. We now need to think about our own knowledge now. So what do we know about these different nutrients? What role do they play in our digestive system? So protein, the role of protein for us to consume it is for growth and repair. Fiber, the reason why we consume fiber is to help with digestion. And if we don't take in fiber, it can lead to constipation. Fat, we take in fat slightly for a source of energy. We also need it for insulation and protection of our organs. However, the problem with taking in too much fat is that it can lead to obesity. And obesity is a risk factor for type 2 diabetes. Carbohydrate, we take in carbohydrate to allow us to be provided with energy. And cholesterol, the problems with cholesterol which we've learned is that it can lead to coronary heart disease, which is narrowing of your artery. So we've looked at our own knowledge now for each of these nutrients. We now need to try and evaluate this properly. So let's go in order, starting with protein. We can say protein is higher in meat burgers. This is needed for growth. Fiber is higher in meat-free burgers, which helps in digestion. A lack of this can lead to constipation. Fat is higher in meat burgers, which can lead to obesity. Obesity is a risk factor for type 2 diabetes. So we've gone 3 points. We've compared higher, higher, higher. We've used our own knowledge, talked about growth, digestion, and obesity. We can add one more just to make sure. So we could talk about carbohydrate. Carbohydrate amounts are similar, which is needed to provide energy. So our final thing that we must do is we need to provide a conclusion. So we can say overall, meat-free burgers are better at providing a healthy diet as they have a suitable amount of all nutrients and are less likely to cause problems such as obesity, diabetes, and coronary heart disease. Typically, when you have an evaluate question, you really want to provide a suitable amount of both sides. So really what you want to do is try and provide 2 pros, 2 cons for each, and as well as your conclusion properly and your comparison properly. Okay, question 3. A student prepared some onion cells. The student viewed the onion cells using a light microscope. This is the method used. Read the method carefully. Reminds us what we need to do. This is a core practical again to do with microscopes. 3.1. Table 4 shows a risk assessment for this experiment. Complete table 4. So it's given us the hazards, both of them, which we need to be aware of in the practical, what these things can do. For one of them, the iodine solution, and then we need to think about how you can minimize the risk. So what will be the risk of a sharp knife being used in the practical? You can cut yourself, person that is using the sharp knife. How would you minimize the risk of using iodine solution? Wear gloves. How can you minimize the risk of using a sharp knife? Well, you're using a sharp knife, so we couldn't write using a blunt knife because it would be changing the knife. How could we minimize the risk of using a sharp knife? Well, we need to cut away from the body. So that minimizes the harm to us. Another thing that could be written to get a mark here is to keep fingers away from blade when cutting. Okay, question 3.2. Give a reason for each of the following steps in the method. A thin layer of onion epidermis is used. We need to have a thin layer so light can pass through the tissue. The other reason why you want a thin layer is so you can see the individual cells. It's important that we say the individual cells because having a thicker layer will make it difficult for you to see separate cells. The way that we would be able to see the cells really is by having iodine solution. However, our next step says iodine solution is added to the onion epidermis. What that's going to allow us to do is allow us to see the parts of the cell. And if you want to give an example of a part of the cell, for example, nucleus, that would be fine. The cover slip is lowered onto the onion epidermis at an angle. Why do we need to put it at an angle? The reason is to prevent air bubbles from forming. Usually when you put something down, face down onto another surface, some air bubbles can get trapped and then you might get confused between what you're looking at. So by putting that angle, it allows this to not be formed. Figure 3 shows what the students saw under the microscope at a magnification of times 400. So figure 3 is labeled, telling us our cells. Magnification again is given to us. It's also labeled the length of a cell. The length of cell Z is in figure 3 is 4.8 centimeters. Calculate the real length of cell Z. Give your answer in micrometers. So we need to recall our magnification equation here. So magnification is equal to image size over actual size. So plugging the numbers in now, what is given to us, the magnification was given to us, that's 400. The image length we have given, which is 4.8 centimeters, and the actual length we don't know. It's what we need to try and work out now. So we would need to rearrange the equation and try and figure out what A is. So we can do that where A is equal to 4.8 over 400. If we do that, we will get a value of 0.012, but this value will be in centimeters. So what we would need to do, because our answer they are asking for us is in micrometers, you would need to convert centimeters to micrometers. And to convert centimeters to micrometers, you would need to multiply by 10,000. If you're unsure about that, converting from centimeters to millimeters is multiplying by 10, and then millimeters to micrometers is multiplying by 1,000. So putting those together, it will be 10,000. So 0.012 multiplied by 10,000 should be coming out with a value of 120 micrometers. Figure 4 shows the student's drawing of Figure 3. Give 2 ways the student could improve the drawing in Figure 4. So how do you draw images from a microscope or scientific images properly? Or what are some of the problems with this? Some of the lines are not connected. So that is one point, connect all lines. The other thing that shouldn't be done when you are drawing is there should be no shading. So we've given our 2 ways, but of course there are a variety of different answers that we can put. What else you could say is when you have a scientific drawing, you want to make sure that the magnification is included, or a scale. A magnification or a scale is included. Onion cells can be seen using an electron microscope. Give 2 ways onion cells would look different when seen using an electron microscope. So we can use our knowledge of how electron microscopes are different to light microscopes here, but the question is asking us how would it look different. So we need to make sure we focus on that when we're answering. So what do we know about our electron microscope? Well, it has a higher resolution and it has a higher magnification. So what does that mean now? If we have our onion cells viewed under an electron microscope, well, it would look bigger. And then that's the job of the magnification being greater. But what about the resolution? Well, what does resolution mean? It means the difference between 2 points. So if you can see 2 different points easier, then that means that you have a higher resolution.
This means that your image will be seen in more detail.
The other difference would be that your image would be black and white, as your light microscope provides color. Question 4. Plants and animals have many defense responses. Table 5 shows some plant defenses. Identify whether each defense is a chemical or a physical response. Tick 1 box in each row. So that means that we're going to get 1 mark if we can get 2 rows. So thick waxy layer on leaf surface, that should be a physical defense. Berries that are poisonous should be a chemical defense, and bark on trees that falls off should again be physical defense. Mimicry is a mechanical adaptation seen in both animals and plants. Figure 5 shows 2 insects, a hornet and the hornet moth. Hornets are insects that sting other animals and cause pain. Hornet moths do not sting other animals. Suggest how mimicry helps the hornet moth survive. So it's 1 mark for a suggest question. So we need to be very careful how we answer this, or else we may lose a mark because of the way that we say, the way that we write. So what do we notice about the hornet and the hornet moth? They look very similar. But as it tells us, the hornet moths don't sting, whereas the hornets are able to sting. So the hornet moth looks similar to the hornet, Why? Because predators may look at the hornet moth, believe it's a hornet, think it's going to sting it, and then it would not eat it. So I want to say the hornet moth looks similar to the hornet to warn off the predators, say they do not eat it. Question 4.3. Adult hornet moths lay eggs that hatch into larvae. Figure 6 shows the larvae of a hornet moth. The larvae of the hornet moth live inside the roots of trees, use the tree roots as a source of food, and cause damage to the tree roots. Explain why a tree might die if the roots of the tree are damaged. 6 marks. So we need to look carefully at these 3 points, as it's going to help us now to try and get the right way of thinking to answer the question. So what can we say about these points? Let's take each one, break it down. So roots of trees. What is the importance of the roots of trees now? Well, they have multiple functions. First one that we can think of from our knowledge of cell biology is that it absorbs water. And we know that that's through osmosis. The other thing that it will do is it will absorb mineral ions, and it will do that through active transport. Using the tree roots as a source of food, well, what does that mean now? Well, what we probably also will happen is the roots will obtain carbohydrates. So it will store the glucose as starch and then cause damage to the tree roots. Well, the other problem with that is the roots are there for anchorage. So if it's not there now, what that means is the tree may just fall. Okay, so let's look at these again now. Absorbing the water for osmosis, absorbing minerals for active transport, glucose storage as starch, and anchorage may fall. So how are these things going to cause a situation where the tree might die now? Well, if there's no or less water, what does that mean? There'll be less photosynthesis. From our bioenergetics now, we know that water is one of the reactants in photosynthesis. Absorbing minerals, the problems with a lack of minerals is some of the glucose combines with nitrates, and then that helps to make amino acids, which helps to make proteins. So if there's less minerals, there may be less nitrate, so there'll be less amino acids or less protein. And then that's going to prevent growth of the tree. If there's less glucose storage of starch, so that means that there may be less respiration, because the plant and the tree needs to respire. Alright, so let's try and formulate our answer now. What we can say is less water absorbed, which means less photosynthesis carried out. What does that mean? This means that there'll be less glucose produced. Okay, so we've said quite a bit to satisfy this first point. Let's see if we can get some more now. Next one. We also said that there's less minerals absorbed, which may mean to less nitrate ions combining with glucose to make amino acids and proteins, so less growth of the plant, of the tree. Then we could say less glucose stored in roots, so less respiration carried out by the tree, so less energy released, which is a similar thing with this top point. This means that less glucose is produced, so that means it would be less respiration, so less energy released also. And then lastly, we can talk about damage to the tree roots. So there'll be less anchorage, so tree may fall. And the other thing that could be an issue with the tree falling is that it may also have less access to light, sunlight, so less absorption of sunlight. Then that would mean that there'll be less photosynthesis also. So the key thing for this question is you need to be trying to link your points together. You need to show that you understand the link between water and photosynthesis. We have done that. You need to show the link between photosynthesis and glucose. We've done that. We need to show the link between minerals and growth, and we've done that with nitrate and proteins. 4.4. The larvae of the hornet moth form when fertilized egg cells divide by mitosis. Describe how mitosis produces two genetically identical cells. Okay, so we're thinking about the process of mitosis only. We know that mitosis is part of the bigger situation, which is the cell cycle, but we only need to focus on that part of the cell cycle, which is mitosis. So what occurs in mitosis? The first thing that needs to happen is your chromosomes duplicate. So you could say your DNA duplicates, you could say the genetic material duplicates, or you could say it's copied, but that should be the first point. What will happen next is the chromosomes will be pulled apart to opposite sides of the cell. Now what will start to happen after the chromosomes are on separate sides of the cell is you'll start to have division occurring, but division of the cytoplasm and the cell membrane, which is called cytokinesis. And then once that has fully happened, we will then be left with two cells. However, why is it producing two genetically identical cells? It's because the chromosomes in both cells are the same. 4.5. The cells which are first formed from the fertilized eggs of the hornet moth are stem cells. Name the process by which these stem cells then form specialized cells. So to form a specialized cell from a stem cell, it goes through one process which is called differentiation. Question 5. Water and carbon dioxide are exchanged between leaves and the atmosphere through pores called stomata. Name the cells that control the opening and closing of the stomata. These are the guard cells. Water moves through a plant in the transpiration stream. 5.2. Describe two differences between the transpiration stream and translocation. So we need to say what transpiration has and what translocation differs to get 1 mark. So transpiration, where is it happening? Transpiration stream occurs in the xylem. Translocation occurs in the phloem. Number 2, we can talk about what is being transported, because they're both transport systems. So transpiration is transporting water. Translocation is transporting sugars. 5.3. Which environmental conditions would cause the rate of transpiration to be greatest in a plant? So how quickly is this water moving and being lost? So our factors that affect the rate of transpiration, 3 of them: temperature, humidity, light intensity are the 3 big ones. So if we look carefully at our options, they're talking about temperature for the first one, and they're talking about humidity for the second one. So if there's an increase in the temperature, there will be an increase in the rate of transpiration. So it cannot be these 2. It can only be left with one of these options. So what will cause a higher rate of transpiration when the temperature is warm is when there is a decrease in the humidity. So it has to be the third one. Figure 7 shows information about the mean width of the stomata in a plant. So we've given our figure. If we analyze the figure carefully, it's given us 2 points on our graph: low atmospheric carbon dioxide and normal conditions. The changes in mean width of the stomata in normal conditions are an advantage to the plant. Explain how. So that means that we need to focus only on our solid line here and then think about how that benefits the plant. So the stomata, what's the importance of the stomata? A stomata has two roles. It is to let gases in and out, and those gases that we should be talking about are oxygen and carbon dioxide. And its second role is to reduce water loss. So our question is 4 marks now. So we probably want to consider both of these functions in our answer. So let's look at the changes, because that's what the question says. At midnight, it remains relatively low, but then when it starts to get closer to midday, it starts to increase, and then at midday, it's at its peak, highest. Then as it starts to get on in the day, it starts to decrease, and then at midnight, it's low again. So we can say that at 0, the stomata is near 0, so it would be closed. And then at the midday point, we can say it's open at its widest. And then again, at midnight, it is closed. So what's the benefit of having this change as it goes from night to midday? Well, what we want to occur at midday is photosynthesis. So what is going to be important in photosynthesis? It will be the light intensity, and that's provided by it being at midday, and it's going to be carbon dioxide, which is provided by the stomata opening and closing. So let's talk about first the changes from midnight to midday then. So at midnight, stomata closed. Why is stomata closed? Because there is less light, and so to prevent water loss. At midday, stomata is open widest. Why is the stomata open widest at midday? As light intensity and the temperature will be at its highest. So to take in more carbon dioxide for photosynthesis. 5.5, the changes in the mean width of the stomata in the low atmospheric carbon dioxide are different from the changes in normal conditions. Explain how the difference helps the plant to survive in low atmospheric carbon dioxide. So let's look at the low atmospheric carbon dioxide graph now and see how that's different. So at midnight it is closed, but you can see that it increases earlier than the normal conditions. Let's go to midday. You can see that the line is higher for low atmospheric carbon dioxide, so that means that the stomata is open wider. Then we go back towards midnight, and we can see that it is open again for a bit longer. So we can say that the stomata are open wider and for longer. And why would that be the case? It is to maximize the intake of carbon dioxide for photosynthesis again.